Hello everybody, today I'm going to talk about qualitative and quantitative analysis of propylene glycol and glycerin in pharmaceutical dosage form. These two excipients are extensively used in pharmaceutical field and cosmetic, and they are being used as a solubilizing agent, a viscosity enhancer, as the moisturizer, and also propylene glycol could be used as a preservative, but the percentage of this excipient in formulation must exceed over 15%. The focus of this slideshow is not about the use of these two excipients, but rather how to determine the amount of them in your formulation. This slideshow is beneficial for people who are doing reverse engineering. There are a lot of methods available to do the work, but I'm going to talk about HPLC and GC. The first method is HPLC with the IR detector, and in this technique the size exclusion column is used. Mobile phase is water, also buffer can be used. In this chromatography we see a very well separation of glycerin from propylene glycol, and also in this particular formulation there were some monosaccharide present that they were all separated. This next method is the HPLC with the UV detector, and the column is the helic column. The mobile phase for this technique is 95% acetonitrile and 5% water, and it works very well for separation of glycerin and propylene glycol from all other excipients. This technique is very suitable for alcohol that have more than 2 OH units, and they don't have a long hydrocarbon back chain. So if it's we are trying to do the separation of fatty alcohol, the retention in the column become very sluggish, and it will elute similar to all other excipients. So this large peak at 5 minutes that we see is actually the all other excipient in the formulation that had very poor retention in the column. The last technique is GC, and that we see very 2 peaks of propylene glycol and glycerin nicely separated, and very much all the GC column will work, DB6 for DB5. They all work well, and it's just the retention time will be either longer or shorter. The HPLC RI technique is suitable for formulation with a very high water content, shampoos, lotions, suspensions, syrup. HPLC helic is suitable for formulation which are having high water content or high fat content, but the formulation should be dispersing well in acetonitrile or 95% acetonitrile. If it doesn't disperse well, so your recovery would be low. GC is suitable for formulation with high fat content like ointment, or formulation that have a lot of white petrolastomes and mineral oil. So you should use heptane and hexane for extraction method. References, there are too many to list, and if you look at the website you will see tons of methods out there that talks about the separation or quantitation of glycerin and propylene glycol. Thank you for watching this video. Have a great time.